Hello, everyone. Hopefully you can hear us. Thank you so much for joining for another Genomics Aotearoa seminar. I guess a couple of things. All the seminars, or most of the seminars, are up on our YouTube channel. So if you want to watch them again because you missed a vital piece of data that's going to solve the problem for your PhD, then please do. They're all on our YouTube channel. We're also, if you've got suggestions for people you would like to give seminars or you'd like to give one yourself, then please get in contact and we'll see what we can do. Today's seminar I've been really looking forward to because it is Richard Newcomb, who is chief scientist or something along that for plant and food. I've known Richard for a long time now and I regard him as one of New Zealand's most interesting scientists. His PhD work transformed the way we think about the evolution of insecticide resistance, and I think pioneered to some extent the use of museum specimens to understand evolutionary processes. He then went on to do a whole load of work on insect olfactory receptors, discovering remarkably that they're inside out, which I still think is one of the most remarkable things about the evolution of a whole class of receptors that are different and strange and odd. On the way, published papers on the genome-wide association studies looking at olfaction in humans as well. So a really remarkable career, and most of that done while still performing all kinds of service roles for plant and food. So I've now lost Richard's talk. It was on, nope, still lost it. So Richard's going to talk to us about genomics and the last 25 years of genomics, and I think he's going to ask you to think about what you think the next 25 years of genomics is going to look like. And this is really timely, given that Genomics Aotearoa is currently looking at developing strategies and thinking about the future and how we sit as an organization in supporting genomics in New Zealand. So Richard, it's a delight to have you. Take it away.
Thanks, Peter. Thanks for those kind words. And many of the things that you mentioned there, I'm going to touch upon in my talk. So you're going to get a little potted history of some of the highlights of my career, and I'm going to use that as a way of talking about the journey that we're all on in terms of genomics. So I will do the usual and share my screen. Is that the right screen?
Yep, that's great.
Great, fantastic. Okay, so the title's actually Where an Interest in Genomes Can Take You. So I want to take you on a little bit of a journey, and then, as Peter said, we'll hopefully have some time at the end to talk about where it might take us for the next 25 years. So yes, it's only been 25 years. The journey that we've been on. So let's advance the slides. Go. So yes, there was a time when before genomics, so BG, and I just want to tell you a little bit about that and what it was like. Some of you might either can't remember anymore or have erased it from your memory, or may not have even been around then. So back when I sort of started my PhD, or pre-genomics, a PhD was sequencing a gene. You could get a PhD from just that. Sequencing often involved a lot of 35S and then a whole lot of fluorescent dyes. They were superseded the 35S, and a lot of us ended up spending most of our time running these sequencing gels and then analyzing the data from those. So it was long, laborious, and sequencing a gene was a pretty good thing. Obviously, PCR was came along at about this time and helped us do that. Back in the 1980s and 90s, the word bioinformatics was only just coming into existence, wasn't even a word. Everyone's very proud of the fact they can script today, but there were no point-and-click software packages back then either, and we all used Unix and a program called GCG. If anyone can remember that, was developed by the University of Wisconsin to analyze gene sequences and protein sequences. So that was all pre-genomics, and that all kind of happened about the same time as I was doing my PhD. So I can tell you how that sort of interfaced with my PhD. And as Peter said, I did my PhD on this thing here, which is the sheep blowfly, Lucilia caprina, which strikes sheep. And so farmers put on these pesticides on the back of the sheep in the hope of killing the maggots of the fly. But unfortunately, the fly developed resistance to these pesticides. And at that time, most of the pesticides were these organophosphate pesticides. And resistance to these pesticides were conferred at a couple of loci using classical genetics, ROP1 and MAL. And then the wonderful thing with this blowfly is you can actually do classical genetics in it. It's got a number of physical markers that have been mapped, and so we were able to do genetic mapping. One of the things that was also noted by researchers before myself was that the resistant flies to the flies that are resistant to these organophosphates, such as diazinon, an esterase seemed to disappear on native page gels and homogenates of the resistant flies. So there was this association between an esterase isozyme and resistance. And so to cut a long story short, if you mapped all these esterases and resistances onto a genetic map of the blowfly, they all co-localized and looked as though there was a cluster of esterase genes that encoded resistance to these traits. Now remember, this is all pre-genomics. So the question became, how the heck are we going to get these genes? Because PCR had only just been invented. Obviously, we'd have really liked to be able to sequence the genome, but that was, at that stage, was just a bit of wishful thinking based on costs and the technologies that we had back then. So we thought we'd be clever and see if we could enlist a closely related species, which is the Drosophila melanogaster, which we all know and love as the model animal or model insect. But would you believe that at that time, Drosophila melanogaster hadn't had its genome sequenced either? So crazily, when I arrived in this lab to do my PhD, about 3 people in the lab had decided they were going to sequence a chunk of the genome, the third chromosome of Drosophila melanogaster, and they actually pulled out and made a physical map with phage, and then were sequencing over this cluster. So when I arrived, I sort of helped finish that off. And then we used the sequence of all those esterases to design redundant primers and pull the genes out of Lucilia caprina. And to cut a long story short, we found that one of those genes actually encoded the E3 isozyme. So you can see here, this is one of those esterases, alpha E7, which we overexpressed in baculovirus, and it has the same mobility as the E3 isozyme from fly homogenates. So we were able to identify that gene going back into the blowfly. And then within that, we're able to identify the mutation, which is in close proximity to the active site serine of the esterase, that confers resistance. And it does that by the mutation gives the enzyme the ability to degrade the pesticide, but at the same time, it loses the ability to hydrolyze the classical substrates that were used on those electrophoretic gels I showed you. So it's lost its carboxylesterase activity, and it's gained phosphatase activity to degrade the pesticides. So that was all very interesting. Got us some fantastic publications, and also it started up a whole industry looking at bioremediation using enzymes that could degrade these pesticides. So that was fantastic. But that was just to illustrate the sort of hard slog. That was a whole lab working in this space for many, many years to get at this gene without a genome. The slightly funny thing is that when the Drosophila genome was then sequenced, we got in touch with Jerry Rubin, who was the person in charge of the Drosophila sequencing project, and said, "Oh, we've already sequenced a bit of the genome. would you like it? And you don't have to do that bit. And they said, oh, to be honest, it would actually be quite difficult for us to work out how to slot it in, and we're using a random approach. And of course, as many of you know, the Drosophila genome was used as the test bed for developing all the technologies that were used in the human genome. So they were using this shotgun approach, which we all use today. And it was the first time that they didn't develop a physical map, but just shotgun cloned the whole genome and then sequenced it. So we'd sequenced a bit of the genome and it wasn't even used for the fly genome. So just as I was doing my PhD, and then soon after, all of the large genomes started to appear. So all the animal and plant genomes were starting to come out. So the first animal genome was Caenorhabditis elegans. It was a physical map sequence. And then, as I said, after that, Drosophila and humans were just using a random shotgun approach. And of course, the important thing there was that they relied on bioinformatics to be able to assemble the genome. So the advent of bioinformatics really was so important to that shotgun technology. And then all the plant genomes started to arrive, first Arabidopsis and then rice, arguably, and as many as a number have argued, is a very, very important genome because it feeds, it's probably the food that feeds the most of human population. So I returned to New Zealand and got a postdoc, and then went to become a lecturer for a year, and then was invited back to, at that stage, Hort Research, because they had some spare money in their bank account and that was burning a hole, and they decided they wanted to get into genomics. So Gavin, Ross and I were sort of put in charge of this money and a whole lot of people to get into genomics. At that stage, it was still probably prohibitively expensive to get into full genome sequencing. So we got into poor man's genomics, which was express sequence tags, which is now what we call RNA-seq. And we commissioned a company at that time called Genesis R&D in Parnell here in Auckland to do EST sequencing for us. And we sequenced lots of ESTs from all of the crop plants in New Zealand. Many were doing this as well, and the other CRIs and the universities were employing Genesis R&D to do this kind of sequencing for them. This was long and laborious, and we didn't really at that time have a way of dealing with the data until someone I'd like to call out came along and developed software for us to do this in a program called BioView. So I'd just like to call out Ross Crowhurst, who did all of this work. This was Ross was a molecular biologist, but he decided he wanted to learn how to do bioinformatics. So at that stage, we had one bioinformatician within the organization, and he was self-trained and started writing all the software to interpret the data being developed by Genesis R&D. The gene resource was incredible for us, and we're able to do lots of work and publish lots of fantastic papers and start to understand a lot of important traits in terms of color and flavor and disease resistance. And that's really was a really important foundation for us. Of course, what came along very quickly after that was full-on genome sequencing. And of course, as everyone knows, that was basically enabled by the advent of next generation sequencing, spearheaded by Illumina, and also a whole range of bioinformatic tools, especially assembly tools, to be able to assemble these short reads into whole genomes, and some tricks like make pair sequencing to enable you to assemble genomes from that data. Of course, now we have long reads, we use genetic maps and all sorts of tricks to assemble whole genomes from short read sequence. So I'm not going to go into that in detail, but just to call out the fact that this was a bit of a heyday for us here, Plant and Food Research. We joined international consortia and we're involved in sequencing the genomes of all of our crops, and also not just our crops, but also pests and pathogens of that. And that it really enhanced our reputation as an organization and got us involved in collaborations that we would never have dreamed of getting into by getting into the space. And it was largely due to the prowess we had in bioinformatics to Ross and his team that he was starting to build after that. It gave us the ability to become part of these international consortia. Okay, so I kind of, as Peter said, I've been really interested in chemosensory science over my career, and looking at all the fantastic genomic resources that are available, and we were developing in our own crops. I was always very jealous of the resources that were available for the human genome. And of course, that's for obvious reasons. So I was always thinking about how I could apply these resources into my interests in chemosensory science. So so I started a project up looking at how I could use some of these resources to understand variation in people's ability to smell different flavors and how this might impact what foods they like to eat. One of the things that was available at that time was there were fantastic affymetric SNP arrays now available to use the variation in the human genome to be able to map some of these genes very quickly. So so I was very keen on looking at variation in olfactory receptor genes in the human genome, and the red bars are olfactory receptor genes. You can see it's a really large family of genes. There's about 480 functional odorant receptor genes and a whole range of pseudogenes, up to about 1,000 genes in our genome. So it's the largest gene family in our genome, spread right across all our chromosomes. So we phenotyped people and their ability to smell a whole range of compounds that were important for various foods that we exported to other countries. So here are some of the compounds here. 1,8-cineol, which is eucalyptus, and that compound is one of the feature compounds of yellow kiwi fruit. 2-heptanone is blue cheese smell, found in some of the cheeses, and it's got also found in banana. As you can see there, all these compounds, they're all sort of found in various foods. And then the last one there, beta-ionone, is a floral note found in fruit. So we looked at some variation in people in terms of their ability to smell these compounds, and we noticed that there was quite a huge variation. Just the x-axis on all these log scales, so there's huge amounts of variation in people in terms of their ability to smell these compounds. And in fact, you can see that some of the distributions there are even bimodal, which is a giveaway for having a genetic basis if you see that that pattern. So isobutyraldehyde here and beta-ionone both have bimodal distributions for these phenotypes. So we were employed these affymetric SNP chips and basically employed a classic GWAS approach to see if we could find genes underlying these traits. And I mean, we didn't have these resources available in the other organisms at that time. I'm just noting that GWAS are commonly deployed now across many different crop species, but these resources were only available for humans at the time. And we got some really strong hits, especially for this last compound here. We got a very significant hit with a gene on chromosome 11 for the genetic basis of detection of beta-ionone. Not only that, these chips were so dense that we were able to basically pinpoint straight down to a cluster of olfactory receptors that underlied the locus. So we could almost go straight down to the gene. We still had some work to do to find the gene, though. And we overexpressed all of these odorant receptors in the cluster. This is the work of Jeremy McRae, a postdoc, in cell lines to find the odorant receptor responsible for detecting beta-ionone. This one here, which is OR5A1. And within it, it had a single nucleotide polymorphism. You can see a theme here. I seem to be able to find single nucleotide polymorphisms, underlying traits at ease. And it's just one nucleotide change underlies that ability to smell beta-ionone or not. And basically, human populations are 50-50 smellers and non-smellers. And we were able to map the position of that single amino acid change onto a predicted structure. We didn't have a structure for olfactory receptors, but there have now just been 2 come out from humans. And then again, this is just looking how that causal variant falls on the phenotype distribution. Okay, so just looking at another application of genomics, metagenomics. I was lucky enough to be a member of the Alan Wilson Centre, a Centre of Research Excellence, while it was funded. And we decided to trial using some of these metagenomic techniques, or at least metabarcoding techniques, out on Little Barrier Island off Auckland. We tried using these to see if we could actually start to assess biodiversity. This was a number of years ago now. It wasn't commonly used, but we were reasonably successful in using it to detect a whole range of different organisms, from bacteria, protists, fungi, and animals. We had some problem. Plants are a bit recalcitrant when it comes to finding good markers to use for metabarcoding. Still are to these days, and we were no exception back there. We tried one marker, but most of what we pulled out were cyanobacteria. So for these kind of techniques to be successful, it's usual that you have good reference data sets. So we went about sequencing. For instance, here we're sequencing CO1 across a whole range of insects that we found out there on Little Barrier. What's really interesting is at the time, the most commonly collected species was a beetle that hadn't even been described. And I think that just tells you something about this. A lot of work to do in New Zealand on biodiversity. We still know so little about what's out there, especially as you go down in organism size. We just know less and less. That gave us huge amounts of ecological data that we could look at. Here's just one example of the kind of things that we could look at. This is just showing scaling with organism size against beta diversity across the island. So we could start to look at all of these classic ecological parameters with this huge data set of a whole range of different organisms that we found out on Aotearoa, Little Barrier. And then more recently, I've had some wonderful collaborations on looking at insect genomes. And this was, again, as Peter pointed out, I've had this long-running piece of research looking at insect odorant receptors. That research now has mainly moved into a startup company called Sentient Bio, who are developing these receptors as biosensors. But I've kind of maintained a kind of interest in this basic science aspect of this work, and started to ask questions about how these receptors originated in insects. Turns out that invertebrates have developed these receptors, and they're completely independent from the receptors we use to smell. So we use G-coupled protein receptors, but insects have co-opted ion channels to become olfactory receptors. And again, it's grown to become the largest gene family within insects, especially the more advanced insects, for flies and the moths. When they were first discovered, it was noted that they had probably evolved from taste receptors, and that there was a receptor here with this arrow in green here called orco, which is a co-receptor, which seems to be required for all of the other odorant receptors to function. And there was a hypothesis that this was probably a co-receptor that bounds to the ligand binding receptor and allowed it to form an ion channel. So we're very interested in when these receptors arose in insects. So they probably, we knew at that time that there were no olfactory receptors in crustaceans, but they were all present in the higher insects. So there was this big gap here in our knowledge in terms of when they might have arisen. And that was largely because we had a genome of a crustacean, Daphnia, and we had genomes of higher insects, but there was very few genomes down here in the base of the arthropods for us to work with. So with Thomas Buckley and others, we set out to sequence a number of these basal insect genomes. We sequenced with Calembula off Little Barrier Island, but we found no olfactory receptors in this beast. Chen Wu, this is her PhD work, and now is working for Genomics Aotearoa, based here at Plant and Food Research. Sequenced the stick insect, native stick insect, and for her PhD, and we certainly found olfactory receptors in the stick insect, including orco. Similarly, this is Victoria Twart's work. Similarly, within Weta, we found olfactory receptors, including an orco. And more recently, we sequenced a giant dragonfly and found only a few olfactory receptors in the dragonfly. And then mayflies, we found more olfactory receptors in mayfly. This is the work of another PhD student who's just recently left Sridevi to go and work in France. We also were able to look at RNA-seq data from silverfish and found olfactory receptors in them. So we filled in this kind of large gap and were able to start to look at where these olfactory receptors may have come from. But what was kind of more intriguing is that we also found that orco was probably not at the base of these odorant receptors, but was probably just one of a number of radiations of olfactory receptors from the gustatory receptors or taste receptors. And there's been quite a few radiations of these olfactory receptors. And one in the higher insects, this orco has become locked in as the co-receptor, but there have probably been many different experiments with this system in these basal insects. So we're pretty intrigued with this. For instance, if you look at mayflies here in this sort of fluoro green, there's a number of different groups of olfactory receptors, clades, within mayflies. So they seem to have held on to all of these different trials of olfactory receptors. And of course, we couldn't have done this without Genomics. Genomics was our base tool for uncovering all of this understanding. So just before we go on to the next topic, I just want to acknowledge all the people that have worked in these areas. As you can imagine, I've skipped over huge amounts of work here. So I'd just like to acknowledge John Oakeshott and Robin Russell for all the blowfly work and being my fantastic PhD supervisors. Well, I'd especially like to acknowledge Ross Crowhurst and all the staff at Hort Research at the time when we started doing ESTs and genomics. As I said, Jeremy McRae, Sarah Yeager and others in the sensory and consumer team for the human genomics work, and Andrew Tolpider and members of the Alan Wilson Centre for the metagenomics, and Thomas and Melissa, and the many talented graduate students who worked on the origin of the insect olfactory receptors. Okay, so that was the formal part looking back, but what I'd really like to do now is think about what we think genomics might look like in the next 25 years up until 2050. And during my talk there, I had a whole lot of headlines in terms of different areas like metagenomics and others that have featured. I just wonder whether we can have a bit of a think about what the headlines might be for the next 25 years. So maybe we can, if I stop sharing, maybe we can think about some. Let's see.
Right. So while Richard gets himself organized, I'm going to do the clapping for you all. So that was a fantastic talk and hit on all the cool things that I think Richard has done. So that was good for me. I guess so I don't know how I guess what you want to try and do is run a discussion session. So I guess there are I think we have the opportunity to have a reasonably good discussion. I see no reason why this has to be done in the chat. So if people want to make a comment or give us some ideas about what you think the future of genomics looks like, feel free to unmute and say something. Otherwise, I'm just going to ask Richard questions about olfactory receptors in insects. So maybe I'll kick some things off. One of the things I think we are moving towards is this sort of genomics at scale. I think you touched on it with your olfactory receptors, right? These are massive families that are present across all of these insects, and the complement of olfactory receptors in each of those insect groups presumably feeds into the biology of those insects, right? So it's crucial to their life histories. And so for us to understand on a broader scale the evolution and the functional biology of those systems, we're actually going to need thousands of genomes, and the ability to have those thousands of genomes all organized in the same way, all annotated in the same way, such that the sort of between genome comparisons on a large scale are actually meaningful. And I guess that's where I see some of the challenges at the moment, is that we make our little bespoke genomes, we make our wetters, and we make our common wasps, and I do it one way and you do it another way. How are you going to get to the point where actually we can harmonize that information so that when I talk about olfactory receptors in a wasp, I know that I'm talking about the same genes, or at least we have looked for them in the same way, so that everything is comparable?
Yeah, I think that's a good point. I think we'll start to see a lot more standardization in the and as we see more pipelines developed, and maybe as we see artificial intelligence used more and more as well in the analysis, that we might get some more standardization and be able to do that en masse. I didn't even have that in my list because I thought that was going to be a kind of the fait accompli, that we'd probably very soon have genomes of just about everything that we would ever work on, because we'll have pipelined a lot of the sequencing and analysis of that. Don't know what others think about that.
I feel like a radio show host. If you've got a question for Richard, feel free to jump on the line. And
well, I'll go to some of the areas that maybe that might generate some discussion.
Yeah.
I mean, I've got about 6 different areas here. I mean, I think another thing that we probably will, I know you've been doing a lot of work on this within GA, but I think the data sovereignty issue is going to be a big one, and working through all of that, the kind of permissions part and the co-innovation and part will, I think, be something that we we'll have to sort out within the next 25 years. I think the introduction of artificial intelligence into analysis and bioinformatics will be a major endeavor over the next 25 years. Already we're starting to see its application, but I mean, I can just see that it would automate so much of what we do in this field, all the way from the genome assembly, everything, and then gene identification right through to function prediction, going through AlphaFold, it will just, I think it will streamline a lot of what we do. The other one for me would be variomics. So we're starting to see that with humans, but I think that that will become just a matter of course for just about any organism that we work with. There's no such thing as a genome. We know that's an artificial construct, that it's actually there's a population of genomes out there, and we don't really, I always remember some of the work on grape, which showed that there was a core genome, and then there's actually there is a real variome in terms of even genes. It's not just variation, SNP variation. It's, some plants have extra genes that other plants don't have, and certainly that happens in the microbial world. So I think variomics is going to become big. I also wonder about personalized genomics and personalized sequencing, and that all of that will be driven by technological advances. We've already seen the advances through nanopore, but as we get more and more technological advances and the ability to personalize sequencing even more and streamline the analysis, we're going to see more and more personalized genomics. I also wonder whether we're going to see ecological genomics. I mean, I talked about metagenomics, but are we actually going to see people studying the ecology or systems biology using genomics? Is that going to be a tool that you use that an ecologist begins to use? Is that possible?
Or even in a environmental monitoring sense, right?
Yeah.
Not just what species are here, but also what functions are.
Yes, I think that functional piece is where we need to go as a discipline.
Yeah.
And then finally, I think we're going to start to really see application both in the economic, social, and environmental areas for genomics. I mean, genomics has been great. We've discovered a lot of things as researchers. We've found out a lot about biology, how things work, but I think we've yet to really see the real explosion of application of that knowledge into the real world. And I think we'll start to see that more and more.
Yeah. So Priscilla says, there's still a need to increase database coverages. I wonder how the big consortium will play and help this in the next years. Can I say something controversial? I'm really kind of disappointed. So I'm good friends with people who are sequencing, the Darwin Tree of Life project, for example. They're going to sequence every species there is in the UK, and I see the point, right? That background information will be huge, and in fact, their current information coming off their sequences is fantastic. But what I worry is that we are doing the easy thing, sequencing genomes instead of understanding them. The big consortium with the big money behind them are doing, let's sequence the genome, throw it out, and then the function is being left behind. And the function is the important bit, right? The function is the bit that's going to help us in an agricultural sense, or help us cope with climate change. So I think that those big consortia are kind of interesting. Patrick Biggs has his hands up, and I know the blood will be rushing from his fingers. So, Patrick, do you want to say something? You see, he's fainted due to the...
Hi, Richard. So I have a couple of things. One, what do you think raw data is going to be like in the future? Because if you think about the variome stuff now, there's a point of view that people don't even bother keeping those reads, that the 99.9999% that don't match, just the ones that do. And then the second thing is, with the rise of AI and more heuristic type approaches, how do you think reproducibility is going to carry that weight? Because by definition under heuristics, you do things twice, you get slightly different results.
Yeah, those are good questions. I mean, I think you've just come across, you've just brought up research questions, haven't you? I mean, I think that's always going to be a big question for bioinformaticians to deal with, in terms of what variation in the sequence data is true, allelic variation that you're seeing out there, or versus sequencing errors and those sorts of things. And I think that's going to be an ongoing research question to be able to decipher that. And then, yeah, you're right in terms of AI. I mean, the real worry is you're going to get AIs AI results, training AIs, and you end up with all sorts of problems. And so, I mean, there are, you know, still huge amounts for us to do there, and there are some concerns. But I'm trusting you to work those out, Patrick.
Oh, thank you very much. Okay. All right. Thanks, Richard.
So Liam says genome modifications, epigenetics, especially with nanopore. I guess that's the point now, that the nanopore, you know, there's a remarkable range of genome modifications that they're going to be able to read, and you know, they are working on proteomics with the same kind of system. Is that the way? I worry that we get into this position where we're measuring everything, and it's really hard to pull out the biological insights in that measurement of everything. But is that, you know, is that where we need to go, actually? I mean, is that the same as Priscilla said, you know, those big sequencing consortiums, sequence everything, sequence all the epigenetic modifications and do proteomics at the same time, have that data available, and then I don't know what, AI to try and pull out some sort of insights?
Yeah, well, I think with many organisms, we are going to have to go into higher order levels. I mean, with some animals and with some beasts, you know, there's probably not so much in that higher order category, so we don't have to go so far to gain insight. But I think with, you know, just the classic example is when everyone was a bit disappointed with how few genes humans had, but then everyone sort of sighed. There's a big sort of sigh of relief when they worked out, well, yes, but there's huge amounts of differential splicing. There's a whole lot of genes we never knew existed, and there's much more higher level control going on in those genomes than there are in other genomes. So yeah, I think, you know, for humans and other for a number of other organisms, we're going to have to go quite up through the levels, because it seems to be much more information going on in higher levels with splicing, you know, modification of nucleotides, all sorts of epigenetics. So yeah, we're probably going to have to.
Yeah, more expense. We have a comment from Josh who says structural variants within species and population genomics have been getting a huge amount of attention lately, largely because of moving away from solely generating genomes through short-read technology. What other big things are just over the horizon of our current technologies? I guess that's a good point, right? A lot of this is technology-driven. What do you know? Is there you've always got your finger on the pulse of what the next big thing coming down the pipeline is. What do you think it is?
I mean, yeah, that's a really good question. I'm not sure I exactly do. There's so many different data types and streams. I wonder whether the next big thing is trying to actually use analytical tools that will actually be able to draw insights across multiple data types, whether that's knowledge graphs or whatever it is. I think we're going to get to the point very soon where individual humans are not going to be able to make those draw those lines across multiple data, and we're going to have to start relying on other kinds of analytical tools to make those inferences for us. I mean, I showed you during these talks things that you know, stuck out like sore thumbs, right? They were just anyone could have seen them. They were so obvious, but really there's probably very few of those kinds of insights, and we're going to have to start digging a lot deeper and using other kinds of tools to find the insights that we need.
Okay, so we've got tons of questions here. So I think Jean Fleming asks, does the future include the use of these data for biosecurity and monitoring of pest or invaded insect species, as in the development of new specific laws? I mean, bloody hope so. I mean, I think there's a crucial problem. But what do you think, Richard? Yeah.
Absolutely. I think there's plenty of application for these and sort of safeguarding our border biosecurity threats. Increasingly, we're going offshore to study those organisms off in the country of origin before they get here, and then developing tests for when they do get here, whether that's a DNA-based test, or pheromones, lures, those sorts of things. So, but absolutely, there's genomics will be part of that.
Yeah, and Lily Barker asks, I think this is a really interesting question. Germline human genome editing and the ethics around this topic. I guess one of the things I do wonder in this space. So we often in New Zealand separate the sort of genomics from the genome editing. That genome editing is a response to, I guess, that's the functional end of things. But we kind of believe that we can do genomics without having to really do the genome editing, but which I think is a bit naive. Sorry, my phone is ringing.
Well, certainly the opposite's true. We can't do editing without genomes because we don't know what we're editing unless we have the genome. So I think the genomes have been essential to our ability to, use editing technology.
Yeah, I just wonder if we do find, variation that is actionable in human genomes, for example, is it the case that we're going to have to go to genome editing, or do you think actually ethically this is something that, so often when we discover a technology that can do good or harm, we use it. There's no question about its use or not. Do you think that genomics really is the kind of technology which, once it points out the sort of changes that might need to be made to improve someone's life, that then there is an ethical, what is the ethics around? I guess not asking what the ethics is, but do you think we can stop? Does the ethics of saying, okay, we don't mess with human germline, is that ever going to stop us in this situation?
I'd like to think we can stop ourselves from doing things, and I just I'm reminded by when restriction enzymes were identified and people worked out they could cut DNA up and put it in plasmids. Actually, the researchers got together and decided how they were going to self-regulate it, and I'd hope that we'd do the same for some of these other technologies that are being developed.
Yeah, that is a story that needs to be told more often, actually. The fact that there was an embargo driven by researchers when recombinant DNA was invented because they wanted to look through the consequences first.
Yeah.
Paul Gardner says, one of the reasons AlphaFold works so well is the large and well-curated protein sequence and structure database. Are there many other large and robust genomic datasets that are AI ready for training and evaluation? Oh, that's a tricky one.
Yeah, well, I hope so, because our gene annotation algorithms are still not particularly good for many organisms, and if we are gonna employ AI to help us with gene annotation, then we are going to need some pretty robust training sets.
Yeah, I've just been looking at some protein annotation tools recently, and I was very underwhelmed with how many of the AI tools were performing. So yeah, I was just wondering if you had any thoughts about data sets that are that well curated at a similar scale.
Well, the most success we've had is basically manually curating genomes with RNA-seq and protein data, and then going through manually and then using them as training sets for either hammers or something else.
Yeah.
It's laborious, but you'd hope that once that got going, but of course then we go back to that risk that Patrick brought up, that and if you let the AIs develop incorrect sets that then are used to train other AIs, you can go horribly wrong.
I think we're almost worse than that, because we've already been down that road, that the function of a gene in Drosophila is used to annotate its function in humans.
Which we have
to annotate its function in sheep, which is used to annotate its function. And at that point, where truth lies, if there is such a thing as truth, is problematic.
Yes, that's true.
All right, Ignacio says, to me, genomics has been discovery focused because everything is new. Sequence your organism of interest and see what looks interesting and then publish. Do you think the future of genomics will move back to more hypothesis testing science? Ignacio is clearly on a Marsden panel. That's
Yeah, I don't know. I think there's room for both. I mean, we've talked. There's still going to be quite a lot of discovery to do. We've talked about these higher level, modified nucleotides, all sorts of other levels that we still don't really understand what's going on there. I think there's still plenty of room for discovery. But I think it's really interesting. I debate this a lot, but working in an applied research institute, I'm always talking about the balance between discovery and research that would lead to application somewhere. So it's that balance, and of course, you've got to keep the investors on the hook, the people that are paying for this stuff, by having the occasional win for them.
Yeah, I'm on the same boat, working in the same institute, thinking about those kind of things a lot. And yeah, to me, I get kind of frustrated sometimes reading those new genome papers, and it's just, they present everything and just say, oh, this is interesting, and call it a day. Anyway, just a thought. Thanks for answering.
So I think this is an interesting point, because I was having a conversation with Mark Blackster, who runs the Darwin Tree of Life project, and they publish all of their genomes in welcome open research. And I don't know if you've got on the treadmill of reviewing for them. It's awful, right? You are getting now 2 or 3 requests, and the papers are effectively, here's the species, isn't it interesting? We sequenced the genome, our pipeline looks good, have a nice day. There's no biological insights. And I think, I guess, when talking with Mark, he feels like, because he wants to increase the scale, 100-fold, 1,000-fold, a genome every 20 minutes kind of thing, this is going to break publishing. It's going to break the peer review process. And I do think that's an interesting challenge. How do we deal with, so I think we need to publish these papers, because something needs to go with the data, so that we know that the data is good, and it needs to be peer reviewed to some extent. But the scale of it is going to become huge, and this doesn't get anywhere near Ignatio's questions about actual, functional hypothesis testing. So how do we deal with that? Do we just have a bucket where we throw genomes in and don't worry about any quality control process?
Or some sort of fast track, I think.
Yeah.
Rather than writing a full paper in a traditional sense.
Yeah, I mean, they're short papers. They're easy to read, but it's interesting that my review responses, they say you have to write a certain number of words, and my review responses are generally things like, yeah, looks good. What do you want from me?
I would have thought that would be a candidate for a pipeline that basically, you stick the raw data in, there's a pipeline, there's an agreed pipeline, there's an agreed set of parameters that you use, there's an agreed set of QA, QC characteristics that must hit these levels of quality, and and that's basically it. Otherwise, you're going to burn out a lot of reviewers. Yeah,
I'm already tired. So Olin asks, and I think this is actually a really interesting question in the context of that talk. He says, do you see an increase in high throughput phenotyping and image analysis?
Yeah, that's a really good question.
And I guess there is high throughput genotyping. Where the hell is that data going? How does that work?
To be honest, to create insights, and a lot of these techniques we're employing, genetic-based techniques, where it might be mapping or whatever, or breeding, or actually the genomic part's becoming relatively straightforward, and even the bioinformatics is not too bad. But actually the phenotyping and the analysis of the phenotype data, the image data, whatever it might be, that's actually becoming the difficult part. It's more bespoke. It's difficult to standardize. There's all sorts of reasons why it's more difficult. It's very, you imagine trying to phenotype what's behind me, these apples. It's not straightforward. So I think that's probably a really big challenge for the field. Moving again, if you want insights in the biology and you want to understand the variome and how the variome translates to variation in phenotype, then phenotyping becomes really, really important.
Yeah, I think that the size of these databases that we're going to need to have. I mean, just looking at the apples behind you, they are apples, I think. The phenotyping of those is huge. Every individual flower, etc., and all of those traits, and all of that information will need to be there with the genomics in the long run.
So we've started to work out how to do high throughput phenotyping in this apple orchard. This is a 2-dimensional array apple orchard. So it's a planar setup. Oh. And we've got cameras around single trees, and we're starting to work out how to do it on a scale as well, of looking at the whole orchard. And they're collecting image data every 2 days down to in high resolution, so we can actually look at development of buds, flowers, and fruit over a growing season. So you can imagine how much data is being collected is just phenomenal.
And then you need to track every single pollinating insect. And Yeah, but I do think that that there are huge insights to be made from that. I don't think this is a law of diminishing returns. I think actually there's a really good chance that this is going to be the next breakthrough.
Can I ask just a follow-up question on that? Do you see space for crowdsourcing any of that phenotyping or data collection?
Cool idea.
I mean, there's a lot of cameras around.
Yeah, I think eventually we probably could, but again, it's like you probably have to set up the initial data sets so the crowdsourced stuff can be mapped onto something. And then of course, there's quite a bit of crowdsourcing going on when it comes to annotation of the image data before it can be used to train an AI, for instance. So that's another sort of crowdsource, although there are companies that will do a lot of that annotation of the images for you.
So we're coming to the top of the hour here. There's one more question from Jean, who says, we're reaching a point where resequencing is the same price as long-term storing the sequencing data. How do you see that going forward?
Yeah, that's going to be really interesting, I think, because there's a lot of these like sensor technologies now which collect data at the periphery of the sensor array, and then they don't actually push all the data down to the to be stored, but they actually do some of the analysis, at the edge, and then push down relevant information down. And maybe genomics might end up going the same way. We won't actually end up storing all of the data, but we'll just store the variants if that's all we're interested in. Again, this is showing my age. When Illumina sequencing first came out, if everyone can remember how it works, basically they take pictures of the fluorescent dyes turning on and off after each round of reaction. The software to analyze that data was originally astrological astrological software, if I get them, you could get the image files if you wanted to with the sequence data. So the image files were 1 terabyte for each image, and so we quickly worked out that there wasn't enough memory in the world to store the image data. And I think there's lots of cases of that. Now we're going to have to work out how to get the information we require out, especially out of image data, because it's just too much of it.
Okay, folks, I think we better leave you to your Friday afternoons. I'd like to thank Richard for a really stimulating conversation after his talk, and also an excellent talk. I still have questions on your talk. I might just have to tell them to you. That was really illuminating, and as I said, timely as we kind of think about the future of genomics Aotearoa. So thank you so much, and thank you for everyone who participated. That was really helpful. I'm hoping, Jess, if you're still there, that actually we can leave the question and answer session on the recording, because I think that's a kind of illuminating set of discussions. So thank you very much, and we will see you again in a couple of weeks. Thank you.
Thank you, everyone.